Okay, so today I'm going to talk about my project that is based on studying central nervous system growth in health and disease, and mostly I'm going to present the work that I done in the laboratory of Elisa Marti during my postdoctoral fellow. So in the lab of Elisa Marti, we are mostly focused on determining the cellular mechanism underlining the correct patterning, growth, and shape of the central nervous system at the early primordium stage that correspond to the developing neural tube. And what we use as a model is a cheek neural tube that has been shown for decades to be an excellent in vivo model to study central nervous system growth in health and disease. So in this slide, what I'm showing you are two transversal sections of the neural tube at different developmental stage. At the left, it's an early stage where you can see neural precursor cell in the ventricular zone receiving signal from the notochord and the floor plates, such as sonic hedgehog, and from the roof plates, such as wind and BNP. And these morphogenes are important for the patterning of the neural precursor cell population that will express different markers, as you can see in this figure. So when this happened, at the same time, the system grow. And this growth is induced by the proliferation of neural precursor cell within each of these domains. And this is further counterbalanced by differentiation of the neural precursor cell into neurons that will delaminate from the ventricular zone to go outside and form these different type of neurons in the mental zone. So embryonic growth of the central nervous system depends on the occurrence of three modes of division. Neuroepithelial cell undergoing interkinetic nuclear migration undergo proliferative PP mode of division to give rise to two progenitor and maintain the stem cell pool. Neurogenic PN division to give rise to one progenitor and one neuron. And neurogenic NN mode of division is when after a division a cell gives rise to two neurons and exhausts the stem cell pool. Also, the rate of proliferation and the type and function of neuron differ all over the rostrocaudal axis of the central nervous system. This mechanism is well conserved. So in the lab, we generate the molecular marker to in vivo monitor the mode of neural precursor cell division. And these fluorescent markers are based on the activity of two promoters. The SOX2 promoter, which will get active once a division will give rise at least to one progenitor, and the TIS21 promoter that will get active when a division will give rise at least to one neuron. And the combination of the two reporter will help us following each of the mode of division with a color code. Then the PP will be in green, the PN in yellow, and the NN in red. Here you can see in vivo live imaging of cell undergoing interkinetic nuclear migration and dividing PP, PN, and NN mode of division with the color code that I mentioned before. So we used this reporter to study the impact of sonic hedgehog signaling on the formation of the motor neuron progenitor domain in the ventral part of the neural tube. And what we found is that when we active hedgehog signaling by electroporation of the receptor smoothened, an active form of the receptor smoothened, we are able to increase symmetric proliferative division. However, when we prematurely inhibit hedgehog signaling by the electroporation of an active form of the patch delta loop of the patch receptor, we are able actually to induce a premature symmetric neurogenic NN mode of division at the expense of PP symmetric division. So what could be the molecular mechanism involved in the control of the mode of division? So here it's very important to highlight that each of these neuroepithelial cells localize in the most apical part the centrosome that is able to generate a slender organelle called the primary cilium. And this primary cilium is really important for the reception of sonic hedgehog signaling that will get secreted into the lumen. When the pathway is active, many components of the sonic hedgehog signaling get localized into the primary cilia, such as the receptor smoothen, the transcription factor Cl3, and we have also the protein kinase A that is really important for the processing and the activation of Gli that will get mostly localized to the centrosome at the base of the cilia. But hedgehog signaling localization at the centrosome will not get only restricted to the basal body of the primary cilia, but what we found also is that also it will get the protein kinase A, for example, get localized to the spindle pole in mitotic neural precursor cell. So what we found is that the protein kinase A get localized symmetrically when neural precursor cell present a high sonic hedgehog activity and undergo proliferative PP mode of division. However, the recruitment of the protein kinase into the spindle pole is asymmetric when the cell do not show any hedgehog activity and the division is neurogenic. So through the transcription analysis, we found that sonic hedgehog is able to control many, the expression of many genes involved in the maturation and the integrity of centrosome, and some also scaffold proteins such as the pericentrin involved in the recruitment of PKA at the spindle pole. So here in this slide, what I would like to highlight is the importance of the cilia centrosome axis in dictating the mode of division. And what I showed you also is that the mode of division is not stochastic. It's an event that is completely controlled by signaling pathway. So hence the control and the balance between the mode of division, the three mode of division is really important. And if any premature neurogenic division occurs, this could exhaust the stem cell pool and could induce brain size abnormalities such as primary microcephaly. And primary microcephaly is a cell division associated disease that has been shown to be genetic. Since from the 20 loci described from now, there are 13 genes to encode to centrosomal protein. And also it could be non-genetic, so like environmental, like in the case of Zika virus infection. So a brief historical introduction of the Zika virus. The Zika virus has been discovered in the Zika forest of Uganda in 1947 in the rhesus monkey. And after, in the 20th century, it spread eastward and went through mutation to form the Asian strain. However, the massive outbreak came with the occurrence of the American strain in Brazil in 2013, when actually the Zika virus were inducing microcephaly and congenital malformation in fetus and in pregnant women. So the Zika is transmitted by the active Aedes mosquito. It's a flaviviridae RNA virus, and it has been shown that a single mutation in the capsid of the Zika American strain made him more virulent and more infectious to neural precursor cells. So in collaboration with a senior postdoc in the laboratory of Nuria de Verdaguer in the structural biology department at the IBMB, we went studying the impact of the proteins translated by the Zika messenger. So the Zika polyprotein is constituted of some proteins that are involved for the formation of the capsid and other non-structural protein classified from NS1 to NS5. So when we electroporate each of these non-structural protein into the cheek, we were able to induce premature differentiation in the case of NS2 and NS5. When we were doing this study, some lab at UPenn in the US published how NS2 could impact the pool of progenitor cell by affecting adherent junction in brain organoid. However, the function of NS5 or the impact of NS5 on neural precursor cell was completely unknown. So that's why we decided to continue with this protein. So we more deeply studied the behavior by using reporter of proliferation like SOX2 and two other reporter of differentiation like NeuroD and tubulin 3. And also we saw again the impact of NS5 on inducing premature differentiation of neural precursor cell. Now, when we look at the subcellular localization of NS5, you can see that it is localized into the nucleus, and most interestingly, also it is associated to the centrosome at the base of the primary cilia. To study what could be the impact of NS5 localization at the base of the primary cilia, we went doing an interactum screening using a human fetal brain cDNA library. And what we found is that from the 21 host protein, 12 protein are associated to the cilia centrosome that you can see here in this illustration, and some of them are also associated to the nuclear envelope. So we choose to focus mostly on three interesting centrosomal protein, which are the CEP164, localized in the distal appendage and involved in xyliogenesis, the centrosomal protein BART, and the ruticin, which is part of this rootlet fiber here, that is important actually for the linking of the primary cilia to the nuclear envelope. So here, a brief recapitulation of ciliogenesis is like ciliogenesis is a high dynamic event that occur in coordination with cell cycle progression. At G1, the length of the cilia is very long, and after it gets shorter to remain as a ciliary remnant at mitosis and get inherited with the mother centrosome to the spindle pole. So we went recapitulating the cilia length in neural precursor cell, as you can see here, by electroporating a ciliary membrane marker, IRL13B. And you can see how the cilia lengths change according to the cell cycle progression of neural precursor cell. However, when we electroporate the NS5, we can see its ability to localize in the centrosome at the base of the cilia. And we can see the resorption of the ciliary structure, which could be defined as microcephaly, as ciliopathy. So here there is a quantification where you can see the ciliogenesis in a control matter. And what we saw is that when the ZKNS5 is only localized within the nucleus, the ciliogenesis occur as in the control. However, when the ZKNS5 localized in the base of the cilia, here we can see a significant shorter cilia length. We went quantifying the centrosome localization of each of the host proteins that I mentioned before. And what we found is that when the ZKNS5 interact to the centrosome, there is a decrease in the amount of the centrosome localization of this protein. So whether this is due by a local degradation or a dislocalization of this protein from the centrosome, this remains a question that we didn't answer. So another event that occurs also in the apical part of neural precursor cell is what we call neural delamination. During neural delamination, there is a kind of actomyosin microtubule ring here, belt that get formed and is involved in the apical restriction and shrinking of the apical pole of neural precursor cells. So then the apical foot is able to detach from the apical surface, and the cell is able to migrate into the mental zone. So to see whether the ZKNS5, since we saw that it is localized in the most apical part also of the neural precursor cell, we were wondering whether it could have an effect at this level. So we did an open book preparation of the neural tube to have an in-phase view of this event. So here, as you can see in the first figure, the centrosome highlighted in red, ZKNS5 in green, and each apical foot is limited by the N-cadherin polarity marker. Okay, so what we can see here is that first the ZKNS5 is localized with the centrosome in the middle of the apical foot. After, both of them migrate laterally, and what we can see afterward is that the ZKNS5 is not specific to the centrosome anymore, but it's spread all over the microtubule belt surrounding the apical foot to also be associated to apical restriction. So we went quantifying the percentage of apical restriction in embryo where we electroporated ZKNS5, and we can see this percentage, this increase in percentage of apical restriction means that ZKNS5 not only induces ciliopathy, but also promotes apical area restriction and premature delamination of neural precursor cells. So what could be the output of affecting such, like this set of cell fate determinants on the mode of division? So to answer this question, we went electroporating ZKNS5 with the T-SEN-SOX2 reporter, and by FACS analysis and by quantifying mitotic cells also in transversal sections, we were able to see that ZKNS5 promote terminal and neurogenic division at the expense of symmetric division, and this could explain somehow the profile of microcephaly that is induced by this virus. So the ZKNS5 is actually composed of two important domains. We have the RNA-dependent RNA polymerase domain that is important for the replication of the virus. And the methyltransferase domain actually is important for the 5' capping of the messenger. for its translation to protein, and also for let's say protecting the messenger against the innate immune system of the host infected body. So what we did is electroporate each of these domains within the cheek neural tube to see which one is involved in premature neurogenesis, and we found that the methyltransferase domain located in the N-terminal is the one involved in premature differentiation. So within the methyltransferase, the laboratory of Nuria Verdaguer and mostly Diego found that the methyltransferase domain is not only involved in, do not only show an enzymatic activity, but also is involved in the oligomerization per se of the NS5 protein. So we went inhibiting the enzymatic activity and the oligomerization capacity of NS5 in this domain to see which let's say, which activity is important for the impact of NS5 on the precursor cell. And we found that when we mimic the oligomerization capacity, we are able to reconstitute a control, a normal activity, let's say, of the TIS21 reporter. When we look at the subcellular localization of these two mutants of Zika NS5, we can see that when we mimic the enzymatic activity of NS5, the protein is still able to go out from the nucleus and translocate to the base of the cilia and induce ciliopathy. And here you can see the quantification. However, when we mimic the oligomerization capacity, the NS5 stay in the nucleus, is not able to go out anymore, and we were able to reconstitute ciliogenesis, as you can see also in the quantification. In collaboration with the Hospital Val d'Hebron, we studied also the effect of Zika infection on ciliogenesis in a post-mortem human Zika infected embryonic brain at gestational week 22. So here you can see this coronal section of the brain, and you can see the microcephaly, how could impact the different compartment of the forebrain. So it was very obvious in comparison to a non-infected human embryonic brain at gestational week 21. So it's very important to mention that at this stage, apical neuroprecursor cells do not exist anymore. So, or they are going into differentiation to form the different cortical layer here on the cortical plate, or they differentiated into ependymal cells that you can see here. And these ependymal cells, they form the ependymal layer facing the cavity of the brain. And the most interesting thing is that these ependymal cells, they form this kind of motile cilia that are really important for the flow of the cerebral spinal fluid, which is really important for a normal homeostasis and toxin washout from the brain. So in the Zika infected brain, we saw that ependymal cells are highly infected, and we can see clearly how the cilia length of motile, the length of the motile cilia is shorter. So this is a quantification that has been done compared to two developmental stages of a normal human embryonic brain at 21 and 23 weeks of development. So down here, what I'm showing you are three different ciliary distribution of three healthy ependymal cells, let's say. And however, in the infected ependymal cells, you can see how cilia are much shorter and disorganized. So now we're continuing with the Zika project, and what we would like to do always with Diego and the master student in the lab, Nicole Elenter, is to resolve the transport of Zika NS5 from the nucleus to the apical or to the basal body of the primary cilia. Here you can see the sequential localization of NS5. You can see how it is in the nucleus after it go out. After with this in-face view, you can see how it translocates all over the apical foot to get localized into the centrosome at the base of the cilia. By cryo-IM, Diego already resolved the structure of rutlethine NS5 interaction. And what he did is he detected the interacting surface of NS5 with rutlethine. because we think that the rootlet fiber is the one responsible for the translocation of NS5 to the base of the cilia. So what he did, Diego, is to try to mutate this three interacting surface, and he found that the three of them are important for the interaction of NS5 with the rootlet fiber. So now with Nicole, what we would like to do is to make live imaging and try to with a reporter of the rootlet fiber and see follow the translocation of NS5 to the base of the cilia, and also introduce this mutation and see whether we are able to stop this anterograde transport and see whether we are able to recuperate ciliogenesis, and resolve ciliopathy. So I would like to thank all the people who get involved in this project, whether from the present and the past member of Elisa Marti's lab. I would like to thank my collaborator, Diego Ferrero and Nuria Verdoguer. And of course, without the imaging facility and Elena Reboyo, we wouldn't be able to do this beautiful images. Thank you very much.